Good morning. My name is Dr. Giribabu, working as a senior lecturer in the Department of Physiology, Faculty of Medicine, University of Malaya, Kuala Lumpur, Malaysia. This is the micro learning video. In this video, I am explaining the mechanism of transport of the substance across the cell membrane. So basically, the transport of substance across the cell membrane via two types. One is passive transport, other one is active transport. Passive transport can be further divided into three types. One is a simple diffusion, osmosis, and facilitated diffusion. Active transport can be divided into the if maybe the transport via pumps. So those are also two types. One is the primary active transport and secondary active transport. If the substance transport via vesicles, it can be divided into two types: exocytosis and endocytosis. Endocytosis and further divided into pinocytosis, phagocytosis, and receptor-mediated transport. So what is passive transport? Passive transport nothing but in the transport any substance there is no requirement of any energy, the molecule moving from higher concentration to lower concentration. The first type is diffusion. The diffusion further divided into two types. One is simple diffusion and facilitated diffusion. In both cases, and simple diffusion and both facilitated diffusion, molecule or substance moving from higher concentration to lower concentration without using any energy. So means the transport substance along the concentration gradient. So it is also called like a downhill movement. So on the simple diffusion, when compared to the facilitated diffusion, on the simple diffusion also molecule moving from the higher concentration to lower concentration without using any transport proteins. Simple example in our body, in our alveoli, so oxygen and carbon dioxide transport via alveoli, so that is also simple diffusion. So come to the facilitated diffusion. The facilitated diffusion also moving from higher concentration to lower concentration the substance, but the transport via transporters or carrier proteins. Simple example, in small intestine, once the food is digested, carbohydrates will be formed. Outside of the lumen, carbohydrates is there. It need to go inside of the lumen, inside of the epithelial cells with the help of the transporters or carrier proteins, that is GLUT2, glucose transporter 2. So now the glucose moving from higher concentration to lower concentration. So simple like a difference is there. Simple diffusion, there is no requirement of any proteins for the transport of the substance. It will be easily move in the lipid soluble plasma membrane. What is active transport? Here, active transport molecule moving from lower concentration to higher concentration. So, when the molecule or substance moving from the lower concentration to higher concentration, so there is a requirement of ATP or requirement of energy. ATP will be hydrolyzed, energy will be producing. So, it is like a uphill movement. So it is also divided into two types. One is primary active transport and other one is secondary active transport. On the primary active transport, energy will be utilized on the primary part. Simple example is sodium potassium pump or sodium potassium ATPase and also calcium pump or SERCA. So I try to explain with the sodium potassium pump how the primary active transport will be working. So you know the primary active transport present on every cell. So it will be transporting the three sodium ions from inside to outside, two potassium ions from outside to inside. So when the movement of occur like a sodium and potassium from inside to outside and outside to inside, so there is a conformation changes required. So the conformation changes happen via when the ATP was hydrolyzed. So the main function of the sodium potassium ATPase, it will be maintain the electronegativity inside this cell. So what is secondary active transport? Here also the molecule moving from lower concentration to higher concentration with the help of another friend like a sodium. Especially mostly the sodium will be helping on the transport the substance from the lower concentration to higher concentration. So simple example like in the small intestine and proximal convoluted tube, we can see sodium glucose transporter SGLT. So look at this picture. Sodium will be when they're going into the lumen, the glucose also entered into the lumen. So here, the glucose molecule are in the lumen is a low concentration, but in the glucose molecule in the epithelial cell is higher concentration. So the molecule or substance moving from lower concentration to higher concentration with the help of another friend like a sodium. So that's why we call like a co-transport. So when the sodium entered into the cell, it will be accumulation will be happen. So it need to transport from the cell. So with the help of sodium potassium pump. Sodium potassium pump here ATP will be utilized so that energy will be generation will be happen. So it will be transport the sodium outside of the cell. So here energy will be utilized. So we can see the secondary active transport like other cells like a thyroid glands like a this is like a NIS sodium ion transporter and digital helium also we can see sodium bile salts also. Next one is vesicular transport. When the molecules like a large molecule, large proteins are transport inside from the cell and outside of the cell, so the vesicular transport is required. So it can be divided into the two types, exocytosis, endocytosis. So exocytosis is nothing but transport of the substance from inside to outside. Best example is digestive enzymes and hormones and neurotransmitters moving from inside of the cell to outside. With so best example, if you see the neurotransmitter when they're transporting from inside to outside, it is transporting with the snail protein. Endocytosis. What is endocytosis? Any substance coming from outside to inside we call like a endocytosis. It can be divided into the phagocytosis and pinocytosis. Phagocytosis also called like a cell eating. Pinocytosis also called like a cell drinking. Phagocytosis best example is like a bacterial entered into the neutrophil. Pinocytosis best example is whenever the soluble proteins entered into the cell called like a pinocytosis. Finally, what is receptor mediator endocytosis? For example, any like a low density lipoprotein or LDL when like attached to the LDL receptors on the hepatocytes. So when they end when they want to attach the LDL receptors, so the below part the form like a clusterin proteins will be formed. Those clusterin proteins form like a page. They pull this substance. So this is like a clusterin mediator transporters. So now we move to the interaction session. So I hope you understand. The first question is what type of transport mechanism is used by the sodium glucose transporter? The second question is what is the main function of sodium potassium ATPase in cell membrane transport? I hope you can answer this one. The first question is what type of transport mechanism is used by the sodium glucose transporter? Is it simple diffusion, passive primary active transport, secondary active transport, and facilitated diffusion? I hope you remember. Answer is secondary active transport. Next one is what is the main function of the sodium potassium ATPase in the cell membrane transport? So answer is A is facilitating diffusion of the sodium and potassium, pumping sodium out and potassium into the cell by using ATP. C is transporting proteins across the membrane. D is increasing the permeability of the cell membrane to ions. Answer is B. Pumping sodium out of the cell membrane and potassium into the cell by using ATP. These are the reference videos and reference books. Later you can watch whenever you have more information. Thank you for your attention.